Protein and amino acid metabolism and catabolism. In this slide set, we will review the topic of protein and amino acid metabolism and catabolism, particularly in the liver and muscle. The liver plays a significant role in protein and amino acid metabolism. It is involved in protein synthesis, amino acid catabolism, and regulates the supply of amino acids to various body tissues. It is important to note that protein levels in the body and in various tissues remain in a constant state of flux, with body proteins constantly breaking down and becoming synthesized or resynthesized. Amino acids undergo processes of deamination or transamination. Additionally, the equivalent of about 30 to 40 grams of protein is excreted each day. Once amino acids are absorbed, they circulate in the blood. Up to several hours after eating, the amino acid concentration in the blood is elevated. A majority of the amino acids in the blood are then picked up by the liver. These amino acids in the liver are integrated into newly synthesized proteins or are catabolized and largely oxidized for energy. Interestingly, the liver derives approximately 50% of its energy from amino acid oxidation. This varies significantly from the body as a whole, which typically derives only about 10 to 15% of its energy from amino acid oxidation. Muscle tissue also takes up amino acids that are circulating in the blood, with the main amino acids being picked up by the muscle, including the branched chain amino acids, which are leucine, isoleucine, and valine. In the muscle, branched chain amino acids are catabolized and oxidized for energy or transaminated to form glutamate or glutamine. And we'll go over these processes in more detail. This diagram reviews the flow of protein and amino acids from intake to absorption from the gut, metabolism, and then excretion. The first column illustrates the path of protein and amino acids from when they enter the mouth to their entry from the stomach into the small intestine and the movement of amino acids from the small intestine into the blood during absorption. The middle column represents protein and amino acid turnover and metabolism. It shows the movement of amino acids from the blood to the uptake by the liver and the muscle, with again the muscle tissue preferentially picking up the branched chain amino acids. The last column demonstrates the excretion of amino acids and shows the removal of nitrogenous waste in the form of urea through urine and sweat. We also see fecal excretion of protein and amino acids which are not absorbed. The metabolism of amino acids and balance of amino acid intake compared to excretion can be represented by the term nitrogen balance. Nitrogen balance is the difference between nitrogen intake and nitrogen loss. Healthy adults do not significantly increase or decrease body protein and remain in nitrogen balance. However, growing children, pregnant women, developing fetus, among other populations characterized by significant increases in tissue mass, exhibit a positive nitrogen balance. So again, these is growing children, pregnant women, or developing fetus would be demonstrating a positive nitrogen balance because they are increasing tissue mass, particularly muscle tissue. Populations characterized by significant tissue loss, particularly loss of muscle tissue, will be in a negative nitrogen balance. Examples of populations that would characteristically be in a negative nitrogen balance would be patients undergoing chemotherapy, burn patients, individuals with anorexia nervosa. Elderly individuals also exhibit a gradual loss of muscle associated with a negative nitrogen balance. So again, examples of individuals in a negative nitrogen balance would be patients undergoing chemotherapy treatment, burn victim patients, individuals with anorexia nervosa, and elderly individuals that are exhibiting a gradual loss of muscle mass. In addition to the primary roles of amino acids in the formation of body proteins, a secondary role of amino acids are in their ability to be transaminated and deaminated to form new amino acids and an alpha-keto acid, with an alpha-keto acid being the carbon skeleton of a previous amino acid. So again, you can see here we have an amino acid. When it's deaminated, its nitrogen group is removed, and then you have its remaining carbon skeleton, which is called an alpha-keto acid. The deaminated carbon skeleton can undergo many processes, including oxidation to form ATP, gluconeogenesis, ketogenesis, fatty acid synthesis, or cholesterol synthesis, among other processes. There are also various other structures whose formation is dependent on one or more amino acids. We will review these processes of transamination and deamination and the secondary roles of amino acids. We will first review the process of transamination and interaction between an amino acid and an alpha-keto acid. And you can see here, transamination involves the transfer of an amino group or that nitrogen group from one amino acid to an alpha-keto acid, which again is the carbon skeleton of a previous amino acid. These transamination reactions are catalyzed by enzymes called aminotransferases. These aminotransferases require the cofactor PLP, which is derived from vitamin B6. You can see another outline and example of a transamination reaction, which involves, as a reactant, an amino acid, an alpha-keto acid carbon skeleton, and then the enzyme, a specific aminotransferase, catalyzes the reaction, which takes the nitrogen group existing originally on the amino acid reactant, and it moves the nitrogen group to the alpha-keto acid. So what was originally the amino acid now is the alpha-keto acid because the nitrogen group has become removed, and what was originally the alpha-keto acid is now an amino acid since it has added the nitrogen group. It is important to note that the aminotransferases catalyzing the transamination reactions are dependent on the compounds involved and are again specific to the reactants. For example, you can see that the transamination reactions involving branched chain amino acids are called the branched chain amino acids aminotransferase. Those transamination reactions involving tyrosine, for example, utilizes the tyrosine aminotransferase, and so on. Here are two examples of transamination reactions. Here's one example, and here's the second example. The first example involves an alanine and alpha-ketoglutarate as reactants. So the initial amino acid is alanine, and the initial alpha-keto acid is alpha-ketoglutarate. You can see the enzyme catalyzing this reaction is alanine aminotransferase, so again specific to the reactant, and you can see the vitamin B6 derived PLP cofactor. This reaction removes the nitrogen group from alanine, transferring the nitrogen group to the alpha-keto acid, to the alpha-ketoglutarate, and after the reaction, the alanine, which has been deaminated, becomes pyruvate, and the alpha-ketoglutarate that now joins with the nitrogen group becomes a new amino acid, which is glutamate. The next reaction involves aspartate, so that's the amino acid reactant, and the alpha-keto acid again is alpha-ketoglutarate. With the specific enzyme, aspartate aminotransferase, and the vitamin B6 PLP cofactor, we see that when the aspartate is deaminated, it becomes oxaloacetate. The nitrogen group that was removed from aspartate is added to alpha-ketoglutarate, and the alpha-ketoglutarate becomes glutamate. Now we will review amino acid deamination, which simply involves the removal of a nitrogen group from an amino acid. These reactions yield an alpha-keto acid and an ammonium ion. Dehydratase or deaminase enzymes catalyze these deamination reactions. You can see here an example of a deamination reaction. Here it's a deamination of threonine. You can see the enzyme threonine dehydratase catalyzes the reaction with the vitamin B6 PLP cofactor. And with removal of the nitrogen group from threonine, we see formation of a new alpha-keto acid, which is alpha-ketobutyrate, and an ammonium ion. Therefore, from these examples, you can see that the deamination or removal of the nitrogen group from threonine yields alpha-ketobutyrate. We also saw in the transamination reactions when alanine was deaminated, or when the nitrogen group was removed, it formed pyruvate. We also see when aspartate was deaminated and when its nitrogen group was removed, it formed oxaloacetate. Like these amino acids, each amino acid yields a specific deamination product. So for example, the deamination product from alanine, again with the removal of the nitrogen group, the carbon skeleton is pyruvate. For aspartate, when the amino group is removed, its deamination product, the carbon skeleton, is oxaloacetate. So each amino acid has a specific deamination product. This figure outlines the deamination products of each amino acid. You can see that some amino acids have more than one catabolism product. For example, aspartate, when deaminated, can be converted to oxaloacetate, or it can be converted to fumarate. As mentioned previously, the carbon skeleton of amino acid deamination can be oxidized to produce ATP. The deamination product affects where each amino acid enters the Krebs cycle to be oxidized. The amino acid catabolism products also determine other fates of the amino acid carbon skeletons. For example, the catabolism product determines if an amino acid is glucogenic and or ketogenic. While most amino acids are glucogenic, there are two amino acids, leucine and lysine, that are converted to only acetyl-CoA or acetoacetate. We mentioned previously that the pyruvate dehydrogenase complex is irreversible, meaning that pyruvate can be converted to acetyl-CoA, but acetyl-CoA cannot be converted to pyruvate. Therefore, leucine and lysine, since they cannot be converted to pyruvate, are not glucogenic. Several amino acids are both glucogenic and ketogenic, including isoleucine, phenylalanine, threonine, tryptophan, and tyrosine. And you can see that each of these are converted to ketogenic precursors, acetyl-CoA or acetoacetate, but also non-ketogenic precursors. For example, you see that isoleucine is converted to acetyl-CoA, but also isoleucine is converted to succinyl-CoA. Here we see a summary of the amino acid catabolism products.